In this lecture, I'm going to attempt to teach you how a muscle actually functions. So, we've spent the last week or so talking about structurally what's happening inside of a muscle fiber, right? So, now we're going to talk about okay, how does this thing actually work? And it's a lot. So, strap in because it's going to be a lot of information coming at you right now. But I'm going to try to break it down and make it as manageable as possible. So let's talk about what's happening on a molecular level inside of a muscle. So the one thing I hope that you've picked up on at this point is that the actual functional unit of a muscle is called a sarcomere. And a sarcomere is essentially two major proteins which are interacting with each other, and that is actin and myosin. But there's two more proteins which I'm going to introduce to you today, which are a big part of this process. And they are called tropomyosin and troponin. And what they do is they provide us the ability to control our muscles. So my hope is that by the end of this lecture, you have a very good understanding of how a muscle fiber actually works. So what we're looking at here in this diagram is really the relationship between actin and myosin, and then troponin and tropomyosin getting involved. So one thing I want to point out to you is myosin. One thing you'll see here is that the myosin fibers actually have heads on them. They have these little heads sticking up, which are called the myosin heads. So that makes sense. And a myosin fiber is actually made up of hundreds and hundreds and hundreds. Actually, they say about 300 myosin fibers. And each one of those myosin fibers has a head on it. And those heads are going to be what attract to the actin fiber in a muscle. But I'm getting a little bit ahead of myself here. So I'm going to flip to this next diagram, and this just gives you a better indication of the relationship between these fibers. So if you go back to when I drew a sarcomere, one thing you hopefully remember is that it has a myosin fiber, and then it's surrounded on both sides by actin fibers. Actin is going to be the thin fiber, myosin is going to be the thick fiber. And it's the relationship between these two fibers which is what makes a muscle function. So let's get into this thing, right? So if you take a look here, what you'll see is the myosin have these individual myosin fibers, but together they make up what we consider to be the myosin fiber. So a single myosin fiber is actually composed of hundreds of individual proteins that bond together, and they make up this very thick myosin fiber. And each of these little heads is what sticks off and attracts to the actin. Now what you'll see with the actin fiber is a much thinner protein. It's actually two that are wrapped around each other in a helix format. So it looks a little bit like DNA. But one thing I want to point out to you is on top of that are these two new proteins which I just mentioned today. Troponin is this little guy that looks kind of like a croissant, right? It's this little protein right here. It's a globular protein. And then we have this thin ribbon-like protein, which is called tropomyosin. And it's a thin ribbon protein which actually covers up the actin fiber. And this is how we control our muscles. So here's the thing about a muscle. A muscle is dumb. It's a dumb organ. What do I mean by that? Well, what I mean by that is it doesn't actually have any control over itself. It responds to a electrical impulse from a neuron that tells it when to contract and when not to contract. So a muscle by itself just sits there, and then when it's engaged, it's going to actually cause the muscle to contract. So what causes this control is the presence of this this tropomyosin and troponin. So let's go a little farther here. So let's talk a little bit about this. So there are not only 4 proteins in a muscle cell, but we're going to focus on 4 proteins. And those 4 proteins are actin and myosin, which are the 2 stars of the show. And then we have troponin and tropomyosin. And these are what allow us to control our muscles. So if you take a look here, what you'll see is actin has these little binding sites on there. So there's little dark dots. You can see they're exposed here and they're not exposed here. So tropomyosin covers up those binding sites. Now, what are the binding sites for? They are binding sites for myosin. So if I go back to this image here, actually, this is a reality situation. Myosin wants nothing more than to come into contact with actin. There's an attraction there. And it's not romance, it's not love, which I sometimes will use that analogy. It's opposite charges, right? So one thing you hopefully know about the world of science is that opposites attract and likes repel. And there's an attraction between this myosin head and this actin fiber. All that myosin head wants to do is to come into contact with that actin. It's the only thing it wants to do. And it can't, and the reason why it can't is because this tropomyosin is blocking it. So if I go back to this image here, you can see in this image, the tropomyosin is covering up those binding sites. So the tropomyosin covers up the binding sites, and the troponin holds the tropomyosin in place. When the troponin moves out of the way and the tropomyosin moves out of the way, that exposes these binding sites for the myosin. So just a little bit of notes here to write down if you're taking notes. The troponin is a globular protein, and what it does is it provides a binding site for calcium. And we're going to talk more about that as we go through this. Troponin has a binding site for calcium. Calcium is extremely important for this entire process. So if there's no calcium, your muscles don't work. And that's why calcium is a big part of your nutritional intake, because it gets used in so many different areas of the body. We store it in our bones for when we need it, but muscles use it to contract. And this is where they use it. The troponin has a site on it for binding calcium. And I'm going to talk more about that in a little bit. Tropomyosin, these are ribbon-like proteins. So they're thin, long proteins, and they cover the site on the actin, which prevents the muscle from actually contracting. So the tropomyosin, imagine it as blocking the actin from being able to contact with the myosin. So troponin is basically the pin that holds tropomyosin in place, and tropomyosin is what's blocking the actin, and it's preventing the myosin head from attaching to the actin. Okay, let's talk about how this actually occurs. So this next bit of notes here is a step-by-step on how a muscle actually contracts. But I think I want to show it to you visually with a diagram first. And let's see how this goes. And hopefully it makes sense, and then we'll go through it. Okay, so this entire process starts with a stimulated muscle. So what are we looking at here? What we're looking at here is we're looking at a muscle fiber. This red line represents the sarcolemma. So this would be the membrane on the muscle. So this would be outside of the muscle over here. This would be inside of the muscle. Now I know my diagram is not anatomically accurate, but it gives you a general idea. This is going to be the motor neuron right here. So what we're looking at here is the motor neuron. What is this attached to? This attaches to the brain. So this would be a motor neuron leaving the brain and going to the muscle. And in here we have our vesicles. So one thing I hope you remember from the neuromuscular junction notes from yesterday is that there is a neurotransmitter inside of this neuron called acetylcholine. And what acetylcholine does is it triggers a reaction. So this whole entire process starts when an impulse travels down the neuron, and we're going to get to how that works in the next unit. And it triggers these vesicles to release their contents. So these vesicles, which have the neurotransmitter in them, they release the neurotransmitter. And what the neurotransmitter does is it attaches to these little protein channels. Here's the thing you need to know about a membrane. In order for things to go in and out of a membrane, they need to go through doorways, because ions can't just pass in and out, and that's how we control our processes. So what happens here is this acetylcholine leaves the neuron, and it attaches to these little channels here. And what these channels are, these are protein channels that let in sodium. So when they're closed, sodium can't get in, and when they're open, sodium can get in. So what acetylcholine does is they open this channel, and what happens now is sodium rushes in. So these little orange dots here represent sodium. So sodium come into the muscle fiber. Now one thing I hope you know about sodium is it's an ion. And ions have a charge. So when these sodiums come in, they actually make the inside of the muscle cell more positive because they have a positive charge. Sodium is Na+, right? It's a positively charged ion. So what ends up happening is this causes an impulse to travel down the muscle. So essentially this electrical impulse crosses over from the neuron into the muscle fiber. And what it does is it triggers a response where there's an electrical impulse which travels down the muscle fiber, and it actually goes down the T-tubule. So there's T-tubules here, and these T-tubules allow the impulse to go deep into the muscle fiber. Now what you see here is you see the sarcoplasmic reticulum. This little circle here represents the cisternae of the sarcoplasmic reticulum. And what's stored in here is calcium. So these little dots here represent calcium. So that's how this whole process starts. Let's move down here. This is supposed to represent the sarcomere. We have our actin protein. This is the blue line. We have our myosin, which is this green one, and we have our myosin heads. So here's the deal. In order for a muscle to contract, the myosin has to come into contact with the actin. And when that happens, the muscle contracts. That's how it works. The actin comes into contact with the myosin. So let me take these little ribbons away. Okay, let me take these away. This is troponin and tropomyosin, but let me remove those. If I take those away, then this muscle will contract, the actin fibers move inwards, and I'll explain how that works in a little bit. So when a muscle fiber contracts, it's pretty simple. The muscle fiber attaches to the actin, the myosin attaches to the actin. As long as ATP is present, it's going to pull these fibers inwards and the muscle's going to get shorter. And then when the muscle stops being stimulated, the myosin detaches and the actin goes back to its original position. And we have ourselves a muscle contraction. But that would be great if that was the end of the story, except we want to do something really important like control our muscles, right? We don't want our muscles just contracting whenever they want to. We want to control them. And that's where this troponin and tropomyosin comes in. So this little red line here represents tropomyosin, and the black dots represent troponin. And troponin holds tropomyosin in place. And what that does is it prevents the myosin from being able to attach. So as long as that is present, this muscle cannot contract. It's not able to do so. And what prevents it is this tropomyosin. So how does this whole thing go down? Well, this whole thing goes down when this impulse travels deep into the muscle fiber, and that triggers the sarcoplasmic reticulum to release its calcium. The calcium floods into the muscle, and here's the thing about troponin. Troponin attaches to calcium, and when calcium attaches to troponin, it changes its position slightly. And when it changes its position slightly, it moves the tropomyosin out of the way. And I'm going to explain that in better detail as we go through. But for right now, what you need to know is the calcium attaches to the troponin and it shifts the tropomyosin out of the way. And when that happens, the muscle fiber, the myosin heads attach to the actin and the muscle contracts. So basically what the calcium does is it gets the tropomyosin and troponin out of the way so this muscle can contract. And then when the muscle's ready to be done contracting, the acetylcholine gets broken down by an enzyme called acetylcholinesterase, and that pulls the calcium back into the sarcoplasmic reticulum. The tropomyosin and troponin go back to their original positions, breaking this cross bridge, which is what you call it when the myosin and actin come into contact, and that muscle relaxes and goes back to its original position. And that whole process is this thing called the sliding filament model. So let me go over the notes that go along with this, and then that'll be it because it's a lot of information. So first of all, the first thing that happens is a muscle fiber gets stimulated by acetylcholine getting released by the motor neuron. So acetylcholine is released by the motor neuron and it goes across the synapse to the muscle. The second thing that happens is the acetylcholine causes the impulse to travel into the muscle fiber. So think of acetylcholine as the thing that triggers this response. So if there's no acetylcholine, there's no muscle response. So acetylcholine crosses that synapse and it triggers the muscle fiber to contract. And it does that by causing the sodium channels to open. The T tubules, or the transverse tubules, carry the impulse deep into the muscle fiber. That causes the sarcoplasmic reticulum to release the calcium. So the calcium gets released by the sarcoplasmic reticulum. So if the calcium never gets released, the muscle never contracts. So at that point, the calcium binds to troponin, which causes tropomyosin to move out of the way, and that exposes the binding sites on the actin filaments. So tropomyosin is blocking the myosin from being able to do its job, but when the calcium comes into the picture, it moves the tropomyosin out of the way, and now the myosin can attach to the actin. And that forms a linkage between the actin and the myosin. The actin filaments move inwards, causing the sarcomere to shorten, and that makes the muscle contract. Now, if you want to relax the muscle, the calcium gets pulled back into the sarcoplasmic reticulum. So that's when the muscle's contracted, it stays contracted. If you've ever had a muscle spasm before or a muscle cramp, you know your muscle can stay contracted. And if that happens, that means the calcium stayed where it was. So to relax that muscle fiber, we got to move the calcium back into the sarcoplasmic reticulum. And then finally, there's an enzyme called acetylcholinesterase, and that decomposes the acetylcholine in the synapse, and that stops the muscle from contracting. That's the final step in this process. Now, I know that was confusing, but don't worry. This is just a basic overview, and now we're going to dive in and really break it down and hopefully understand it. But those are the major steps of this process. Thank you very much.